In the previous lesson on cancer, we just did an introduction. We learned that cancer cells arise from normal body cells that undergo several kinds of mutation, and the mutations happening in certain genes that are involved in making proteins that control the cell cycle. Those mutations then will create a cell that no longer responds normally to chemical signals from neighbors, and it keeps on dividing uncontrollably. So in this lesson, we'll take a look at a very sort of oversimplified story about how altered proteins, how dysfunctional proteins are involved in cancer. This will be our cell, and first we'll look at how a normal cell responds to chemical signals from its neighbors. Now, to detect signals from the neighbors, cells have proteins on the membrane of the cell. So here's a receptor protein, and its job is to detect a chemical signal from a neighboring cell, like a growth factor. Now, this chemical signal will bind, just so, like a key fitting into a lock. That growth factor will bind to this particular receptor. There are many different kinds of receptors on the surface of a cell. And when that growth factor binds to this receptor, something inside the cell membrane is going to happen. You can see this little blue part of the protein down here is connected to this red protein, this pink relay protein. Now, when this relay protein is stuck to the receptor, to the blue receptor, it is inactive. But we're going to see that the binding of this growth factor is going to trigger a change in the receptor protein, and this pink relay protein will become active. So let's see what that looks like. So here we see the idea here is when this growth factor binds to the receptor changes its shape, and the pink relay protein detaches, and now it becomes active. Now, what is this relay protein going to do? Well, we're going to show that it's going to influence events in the nucleus. So what it's going to do is trigger DNA duplication. Now, this is a complicated process here, and we're leaving out a lot of details. But the basic idea is that this relay protein now is, in a sense, signaling the nucleus that it's time to divide. Right? The detection of the growth factor from neighboring cells is now that message is being sent to the nucleus. The nucleus now will prepare for cell division. Well, prior to cell division, you need to duplicate your DNA. So the cell is going to be preparing for mitosis. And it's only preparing for mitosis when it has detected the growth factor. If there's no growth factor, this relay protein would not be active, and it would not be triggering DNA duplication. The cell would not divide. Now let's see what happens in a cell that has acquired a mutation. So here we're going to start with the idea that the cell has suffered a mutation. Now, a mutation, again, is a change in the DNA sequence. And let's now imagine that we're in the gene for the receptor protein itself. So this is the receptor gene. This is the recipe to build this protein up here. Now, if we have a mutation in the DNA sequence, it means the messenger RNA copy will carry that mutation as well, so that change. And here we see the messenger RNA hooked up to the ribosome, and we see amino acids being stitched together in the sequence specified by the messenger RNA. Well, because of mutation, because of a change in perhaps a letter here, we might have a different amino acid at this location. And we know the amino acid sequence determines how it folds up. So if you have a different amino acid here, it might influence how it folds up. And sure enough, there's a different shape. Notice there's no little piece sticking out here. So here we see the receptor is a different shape than the normal receptor. And just as a reminder, the normal receptor had that little blue piece coming out here that the relay protein could stick to. So what we have then is a situation where this receptor may be in such a state that it cannot bind the relay protein. In other words, the relay protein can't stick to it. And remember, when the relay protein is stuck to the receptor, it's inactive. Well, if it can't bind, it's constantly active. So the idea then is this relay protein is just always going to be in the on condition, and it's always going to be triggering the cell to prepare for mitosis. It's always going to be sending the signal that, hey, you got to duplicate the DNA and get ready for mitosis. So as a consequence then, the cell is always preparing for mitosis. A faulty receptor protein, which means that the relay protein is now constantly in the active state, that leads to a condition where the cell itself is always going to be preparing for mitosis. So we see here then how mutations in certain kinds of genes, the genes that are involved in regulating the cell cycle, mutations can create faulty proteins. These proteins, because they're faulty, are messing up the normal kind of signaling of the cell. And now this cell is going to keep on dividing even when there's no growth factor present. So the cell no longer, in a sense, is listening to its neighbors. The nucleus of this cell is just always getting the signal to divide. Now, I should mention here there are many different proteins that are involved in controlling the cell cycle. So, for example, while here in this example we put the mutation in the receptor gene, we could also have a mutation in the gene that specifies the relay protein. So we can imagine a relay protein that no longer sticks to the receptor, that's just constantly on because it has a different shape. There are many different kinds of genes that make proteins that are kind of running this system here. And so cancer results from mutations, multiple mutations in these different kinds of genes that control the cell cycle. And this is just a sort of a simplified version, but it illustrates this point that cells are like little machines. And when the parts are working properly, the whole machine does what it's supposed to do. But if the cell starts to make faulty parts, then you get abnormalities. And cancer is one of these lethal abnormalities.